All right, I think we can start. I already also launched the live session on the YouTube. So hello everyone on both sides of the internet, and welcome to our small informal lecture on navigating the proteomics community. I'm pleased to welcome Julian Ushkoheit. He is the professor at the Ruhr University Bochum and the leader of the medical bioinformatics group. He led and contributed to many software, including proteomics, and is a prominent contributor to proteomics and bioinformatics communities. And so today we're going to talk about several organizations. And if I may ask you, Julian, to go next. I don't see the slide changing. Right, thank you. So we'll talk about several of them who have their main aim focused on two things. One is the development of proteomics worldwide, and then support of the scientists and everyone around who is pushing the field forward. And one of the largest of those is a human proteome organization. Next, please. Thanks. So from the large, it is a huge organization, and as any group of people, it needs its leaders. There are up to 10 people who take on several leadership positions with their different responsibilities, and then the organization branches into representatives of the initiatives, regional representatives from Europe, Africa, Asia, Oceania, and Americas. And also it includes not only scientists, but the delegates from the industry who can advise and push forward field even faster. Next. So more downstream from the HUPO tree, there are several committees who take on different functions, and I want to emphasize that at least from my best of knowledge, many of them have one to several young career researchers members, among which me and also many other PhD, postdoc, and even master students who contribute to this organization. Next. And then the initiatives who focus on the more of the scientific side than education or support. Julian will talk in detail about Proteome Standard Initiative a bit later. Next. So HUPO has several media representations. One is the website, which is renewed for each year. Then I would recommend you to follow them on LinkedIn to see the updates throughout the year. And also many of the initiatives are recorded in terms of webinars, small courses like Proteomics 101. And they also take interviews from prominent scientists, which is really interesting to see and understand how the field works. Next. Right. So I will focus today on early career researchers' contributions to the Congress organization, and then Janelle will talk about PSI. Next. So maybe the biggest event that HUPO leads is the World Yearly Congress. In 2025, it had around 2,000 attendees, which is huge. The Congress takes place iteratively in Americas, Europe, and Asia Oceania. And fortunately, it also provides support to young researchers in form of travel awards with up to $1,000 US. When you apply, you need to form your CV, personal statement with your motivation to attend, and the proof of status as student or postdoc. Next. So this is the Congress is around 5 days long, and it's pure science from morning to the late evening, if not next day. Talking in numbers, in 2025 there were 30 scientific sessions with 4 to 5 going in parallel in neighboring rooms, which were always filled. There were 21 industry lunches, which included the talks from the scientists who were who led their research using the several industry representatives' products, and also there were several science development meetings like Bioinformatics Hub, which, to be honest, was my favorite part of the conference, where people could sit in circles and discuss what we can do to advance proteomics, bioinformatics, and make it accessible to everyone. And then there are plenty of occasions for the network. The Hupo Gala evening was the thematical gathering in the style of 20s, I believe, with nice food and dancing. And then you can meet people during several early career researchers organized events, and talking to commission editors, to her journey of mothers in proteomics and science in general, and many more. Next. Right. So the early career researchers events during the Congress are led by a corresponding committee. You can follow the website page to check out for details. Next. One of the most interesting events are mentoring sessions. There are 3 around 1-hour sessions during the Congress where more experienced researchers, industry representatives, like editors, come and talk and give advices to the early career researchers, which in turn have their issues, questions, and situations which they want to resolve. In 2025, there were sessions on what to do after your PhD. Then how to strategize your publication plan to get the most from your research, and also how to communicate in interdisciplinary settings, which is exactly what proteomics is about. Next. The ECRs have also their separate networking night event. As in 2025, the Congress was in Canada, in Toronto. The thematic was, of course, the national sport hockey, where everyone could try and throw a ball with a stick. But there was also the bingo cards events where you could go around the room and talk to people you never saw before, but they actually came from different parts of the world and they do completely different things. That's exciting to meet new people. Next. And then ECR committee is all about supporting early career researchers, and something that we really need is the lines in CV to show that we develop our skills and we succeed in it, and many people recognize our our good achievements. And among those is a 3-minute thesis competition where ECRs present their research for lay audience in one slide in just 3 minutes. That's a great exercise, to be honest. Then poster competition, in which I also contributed as organizer last year, where a student will present their posters to several judges during poster sessions. They will get evaluated, and then the best presentation so will get a prize. And manuscript competition, the same idea, but you provide also your publication and scientific presentation. As a result, depending on the competition, top 1 or top 3 ECRs will get the cash prize, certificates, and recognition on the HUPO website. Next. Thanks. So why to become the part of the ECR committee member to drive the organization of this event? Next. Well, if this picture doesn't convince you, I don't know what to tell. There's great perks during the conference, but I'm kidding. Actually, the main reason is that you meet awesome people who are really motivated in many terms in both of science and driving the community, which is really inspiring to continue your own journey. And we know science is pretty hard to do it alone. Then you can also get several opportunities, like you can chair a session on the international conference. It's a great experience to learn how to drive the discussion. You can also get the discount to the conference registration, which helps you to come and present your own research. And then, of course, whenever you apply for scholarships, you're also evaluated as on your leadership abilities, and whenever you volunteer in such organizations, this is a big plus, and you get more chances to get financial support. Next. So if you're already interested, you can become part of ECR. One of the requirements is to be HUPO member, and for low-income countries, it is $30 for all 3 years, so it's just one payment, and eventually you get a great outcome for not a lot of contribution, and then you just need to write to the committee co-chairs, and here's an email. Now I will transfer the microphone to Julian.
So first, welcome from my side, and thanks for having me here. And as a first comment, I would say that I definitely would not be here without being at least part of some of these communities we are talking about right now. So HUPO is a great community, and one part I'm active, basically since I was a PhD student. I was pushed into this initiative a bit by my PI back then, was the, where's my cursor? Was the HUPO proteomics standards initiative. So this, in a first moment, might be a bit dry, but it's a really, really great community to work there. And it performs a global community effort to maintain community-driven standards for proteomics and MS-based data formats. So it's, if you ever worked with MZML before, MZDentML, these formats were really designed there and are maintained there and updated there. And everything like this. Back then, we wanted to have the human readable, but right now we also created a new format, which is the MZPeaks, which will be a modern vendor independent spectrum file format. So it's still in the process of being developed. If you are into this, just feel free to join. We also have a quality control format, which basically is published already, which will hopefully help in just providing or distributing quality control measurements of all mass spectrometry files. And they also provide controlled vocabularies to make this readable in a wider format. And on the right, I have a picture which was from last year, January, I guess it was. It was a meeting of the MCQC group, and there's also me inside there and all the other partners. And this is the guys who developed this MCQC format for the last couple of years and are still doing right now. So the process of the HUPO PSI is that every work is done in subgroups, each one for one topic. There's actually not only computational topics or standards for file formats, but also standards for how to report data not only data analysis, but also data measuring and so on. And if you're interested, you can just check out the homepage which is shown down there, and join any of the group. Everyone's welcome, and you can maybe just join for some sessions. But there's also another opportunity, a great one, because we have yearly spring meetings. So each year, sometimes around spring. The last one was just in Rome last month in May, but sometimes it's also already in February. We have a community get-together and perform 3 to 4 days of really working on the topics you like to work on. So usually you go into one of these groups and work on this format, on this concept. There's also a AI readiness group right now, which just tries to figure out how the data or what we need to make really all the MS data AI ready. And up here you see the group, what it looked like this spring in Rome. And the next meeting is probably in Antwerp. It will definitely be in spring 27. The only time it did not take place was in 2020 for obvious reasons. And new since last year is also that we have poster presentations, especially for young career scientists, and also one tutorial or introduction days where you can really just learn what we are doing and what was done in the last years, and then choose your topic and join. So it's a really good opportunity to get into these projects. Then I will go on with the next smaller group, let's phrase it like this, after HUPO, which would be HUPAR, which is European Proteomics Association. So it's basically the European pendant of the HUPO, and it is not really an own organization, but it's a federation of the national proteomics societies. So each European nation or country has its own national proteomics society, and they together form the HUPAR. And there's also one annual conference. This year, actually, it will be in Germany, in Würzburg, from the 29th of November to the 3rd of December. And we have the typical things there, like posters, tutorial days before the conference, and there are also several ECR meetings. And the HUPAR, besides organizing these conferences, works in initiatives, and especially interesting for you, I guess, would be the YPIC and the UBIC-MS, which I will show you in the next slides. But also just joining the conferences, besides having great science, obviously, it's a lot of fun. Down there is a picture where we actually had a sandcastle competition. So we had the conference in this nice building up there, and it was next to the beach, and then suddenly we had to build sandcastles. In the beginning, everyone was like, nah, I don't build a sandcastle, and then everyone built it, obviously. So it was great fun. So one of these initiatives is the YPIC, the Young Proteomics Investigator Club, which just turned 10 years now. And this is really the ECR club of the HUPAR, basically. So if you are a young scientist in proteomic, you can join this one. So it has its own meetings, but it also organizes special sessions during the conference or on other opportunities for career mentoring meet the expert or just in proteomics introduction if you're new to proteomics. But it also organizes regular webinars and challenges. Challenges are very interesting. So usually you get a sample and should find out what actually was in there or measure it and get the best results of this, which is really fun if you have no clue how to do de novo or something like this. That's how I learned it. And the YPIC also offers one competitive student proteomics fund. I think it was for the last 2 years now, so probably it is yearly now. I don't know how fixed it is yet, but I think it will be around again. The next initiative, where I'm also a member since the founding, is the UBIC-MS, the European Bioinformatics Community for Mass Spectrometry. And this is aims to build and support community of bioinformaticians in computational mass spectrometry. So we really want to be one community and provide online platforms for computational mass spectrometry. So it's not only proteomics, but all flavors of mass spectrometry. We organize international winter schools, which are basically conferences. We organize developers meetings, which are hackathons and are also branded hackathons now. They are not named developer meetings anymore. But we also organize educational sessions and seminars on scientific hubs on larger conferences like the UPA. And we also create some community-driven projects. So we have some larger projects. I won't show them here, but we have, for example, the Proteo Bench, which you might have heard of. Otherwise, you can just Google for it and have a look at it. So it's a really great project. It's the biggest one which took off from now. And we are working really open. So one thing is that we are basically have a horizontal organizational structure, as we name ourselves. So basically, we have no hierarchy. We do have 2 representatives, which is Louise and me right now. So we are representatives for 2 years. And basically, the only reason why we have this is that UPA wants persons to speak to and don't want to write to a mail group. Then we have the UBIC-MS members, which basically is everyone who wants to be a member. And then we have the organizational members, which are the ones which organize right now the conference for the next time. So it's not really that we are voted members or something, besides for the representatives, but otherwise we just work together. We have monthly online meetings where everyone can join. And we have yearly in-person meetings, one organizational meeting, and then the hackathons and the conferences. So if you want to join, you can just scan this code or go to the UBIC-MS or become a member. And basically, we are working with Slack. If you join our channel, just join the general. You will hear some things. Just join the monthly meetings, and hear what's organized. And if you want to organize as well, or just be in the group and hear what's going on, feel free to join. As I said, we have the biannual winter schools, and the next one will probably be around March 2028, and it probably will be in France, so it's not decided yet because it's so far away. But the next hackathon will definitely be in end of January in Sweden. And also an organizational meeting will be in Ghent in Belgium in September. And for the hackathon, we are also right now, so the submission for hackathon projects is open right now. You can just go on our GitHub page, I didn't put it here, and propose a hackathon. If it's selected, then you would have a project on this hackathon. Next one, a bit of another topic we wanted to show here is the so-called International Society for Computational Biology, which is the largest community for bioinformaticians worldwide. So it's like HUPO for proteomics, this is for bioinformatics or computational biology. It is really big and it has lots of communities of special interest, which are COSIs and which are basically the same as SIGs, also special interest groups. And the most interesting for you probably would be CompMS, which also hosts own sessions. And I think that most of the ones who host the Bioinformatic Hub at HUPO and also at the ISMB conference, they are from this CompMS group. So there are several synergies there. You can join the CompMS as well just by joining the Slack. I put a link here. And there's one annual conference of the ISTB, which this year is in Washington, D.C., as far as I know. It's called the ISMB. And every other year, the ISMB is together with the ECCB, which is the European Conference for Computational Biology. And this one is also taking place each year, but every other year together with the ISMB. And then it's a real large conference. As a computational mass spectrometrist, you are basically a total niche there, but you will wonder how many bioinformaticians are running around there. Basically, I think the last one was 1,300 persons or something. It's massive. But for the ISTB, there are also many other regional or topic-specific conferences and events going on around the year. So maybe just have a look at their webpage and see what's going on there. And usually also for each of the conferences, you can get travel grants. I forgot to put this in here, but they usually provide some. With this, I will go back to Europe a bit and show you the Elixir Europe. Elixir Europe is the European bioinformatics infrastructure for life sciences. It's started off to be a community of bioinformaticians of Europe. It is government funded by the nations. And so they just decided that it would be great to work together in Europe a bit more on this for the bioinformaticians, and that's how they were founded. Elixir itself works on several levels. First, there are the nodes, which is one node per country participating, and then we have the platforms, which is the compute, the data, tools, interoperability, and training platform. And you can work on these groups already, or you can also work in one of the communities, which are even many, many more. I just showed some here, which might be of interest for you. There's the Galaxy community, the metabolomics community, the proteomics community, but there are also a lot of others. And these communities are, they are really topic specific and try to advance the field a bit. So most of the ones in the proteomics community are also known from HUPO or UPA events and so on. Elixir is obviously great for networking. It is basically founded to be a network and for networking. So it's really great to learn meet new persons there. And it also has several events which are not exclusive for Elixir members, like the tutorials and training events which take place throughout the year. If you join, just have a look at the Elixir homepage or join also the Slack if you like. You can stay informed there. And there's one yearly big event, which is the Biohackathon. And there you meet with 150 persons, I guess, and just hack for 1 week on one project you decided. I think the projects are decided already, but the submission for participants will open soon. And it will be in November in Barcelona. And these are just 2 pictures from the last hackathon, which was close to Berlin, which was also really fun. Okay, with this I will come to something a bit different, but you might have heard about Proteome Exchange as well. And Proteome Exchange is a consortium which provides global standard data submission to repositories. Sounds a bit dry again, but if you think of that you just are writing on a manuscript or even want to publish this now, then usually you have to upload your data somewhere. And this is exactly what Proteome Exchange is for, to upload your data for the publication. But also, if you have a new tool or pipeline you want to benchmark on some open data, then you can download the data there. You can browse for data which fits to your specific topic, or just download anything and test your data with this and show that it also works with open data, not only with your institute's data. And obviously, it's also a great repository right now to train machine learning algorithms. So you have terabytes of data there to train your IDs, basically. And as I said, it's the institutes are around the globe. I will show them a bit more in detail on the next slide. What I also wanted to say is that besides just uploading the data or storing the data in repositories, Proteome Exchange also has some partners which reprocess the data themselves and give them in a harmonized, standardized way back. So just to identify or quantify the data in a standard way of the uploaded files. So the big repositories of Proteome Exchange are listed here. And depending on where you are or where you want to upload something, for Europe, you would usually use Pride, which also is still the largest worldwide repository. And you don't have any, you need not many metadata to upload something there. You just need your raw files, put some information with this and upload the data. If you can give some more information, that would be great, especially if you later on want to reprocess this data. But if you upload the data there, then you usually also give a link if you want to publish your work later on, and the reviewer can get it there, or someone interested later and see that everything was okay and also reprocess the data. Then in the USA, we have 3 repositories. The real big one there is Massive, which almost is as large as Pride now, and serves the same purpose as Pride. It's just for uploading all mass spectrometry data. Then we also have Peptide Atlas. This is a bit different because it's usually only for SRM or other targeted methods. And we also have Panorama Web, which is even another aspect. It was actually invented for DIA repositories, but also for other targeted. And I think you can also upload DDA right now. On the other side of the globe, we have a repository in Japan, which is JPost. This one is hosted by multiple institutes and is their regional repository. And in China, there's also iProx, which is at the National Center for Protein Sciences, but is also a national effort there. And why do we actually have so many? Because still it's, we're uploading terabytes of data there now, and sometimes this can take quite some time, depending on where you are. I don't know how it might be for you with the connections to these, but for me, especially Massive, is not that quick. And when I was visiting South Africa, accessing any of them was so slow, so that actually they were complaining about not having a repository yet. With this, I will come to my last slide, which is just a little comparison of 3 databases for proteomics, which just serve different aims. So if you ever wondered why they actually exist or where you could get which information best, I wanted to highlight here UniProt, Peptide Atlas, and Protein Atlas. So if you ever want to look for your sequences for, say, identification or quantification of mass spectrometrical shotgun data, or just large data, more targeted data, then UniProt is your way to go. There you can download the annotated protein sequences of basically all species, obviously also very good annotated human data, and it consists of the Swiss prot and also of the automatically annotated Trample part. And depending on how good your data should be, you can download it there. It also cross-references many other sequence databases. It has lots of information, for example, about variants, isoforms, PTMs, and so on. If you rather work on targeted examples, maybe you want rather go to Peptide Atlas, because they provide you with what was measured before. So basically, they can tell you which peptide was found in which measurement before and how the settings were there. It helps you basically to create maybe even a library, but also your target windows and so on. Which you need to create to create SRM or MRM samples. All the data there is reprocessed, and so it's one of the proteomics change reprocessing pipelines, and so it should be standardized and should be of good quality. And then the Human Protein Atlas is even another aspect of proteomics. So it's usually not that much used for mass spectrometrical proteomics, but it's rather for antibody-based proteomics. So if you have your protein of interest and you want to get an antibody, for example, or to fish out something, or just for imaging reasons or whatever, you can go there, and the proteins are also linked. You can basically, so you will get information about the antibodies, and as far as I know, you can also order them there if you're really interested into them. Okay, with this, I give back to Valeria for the last slide.
Yes, thank you very much, Julian. So you might have noticed already that there's various initiatives, and everyone can find something that suits them. You can connect with them online, or you can join offline for fun initiatives. You can go in with your head deep into bioinformatics, or if your heart is still around experimental science, you will find your place. And Julian is a great example of a person who is contributing and participating in many organizations at once. And this is what actually helps to connect all of them and communicate together. And the early career researchers are very, very welcomed. With that, I want to conclude the the lecture part itself. And we can move to the discussion. You can raise your questions. In case it's challenging for you to do it in English, I can translate from Ukrainian. And maybe just before, I want to give you the homework, and you can decide on your own what you want to do. So you can create the Proteomics Society of Ukraine and join EUPA. Or maybe you want to do Elixir Biohackathon next year. That's also a great opportunity. Well, it's a creative homework. Okay, actually I'm kidding, but it's really for you to start thinking about what interests you among organizations you learned today, and if you want to come and at least explore how it all works, you can just join and look, and then see what you can bring yourself. Thanks for your attention, and does anyone have questions, concerns, complaints? Yes, hello.
So, if I'm a bachelor student, where it would be better for me to start in proteomics? Which organization will
Julian,
will be more suitable for me?
Julian, would you like to answer?
So, if you are a bioinformatics student, then I would guess UBKMS, to be honest. Not only to advertise it, but joining there is always good. But also if you have the opportunity to go to one of these conferences, this would also just be a good thing, I guess. Just going somewhere, joining somewhere, and then joining the bioinformatics hubs there maybe, and I guess many persons of several initiatives would be around.
Thank you.
I would say that HUPO-ICR can also accommodate bachelor students. It might be a little bit more difficult, but you can contribute into organization of the conference remotely, and then once you have results, you can join yourself.
Thank you.
Until people are thinking on the question, I remembered one of the aspects of the community gatherings is that sometimes they end up with scientific publication, right, Julian?
Yes. So that is something we always try to push or try to do. So if there's a project going on and you joined it, usually in the end a publication comes out, which obviously helps you in to get to be seen in the community, and also drive your ideas forward. And really, usually in all of these organizations or initiatives, everyone is really welcome. So it's not that you should be, I don't know, top grade PhD or something. Just join and be part of the community and help. That's what it's for.
And I think I saw some of the publications end up being submitted and published in journals, and some are available like on Zenodo or similar repositories.
Mostly, yeah. So if you look at the UBKMS ones, yes. So most of them we try to publish. You obviously are open scientists, so we put them on preprint servers first. But there were also some efforts which, in the end, we deemed to be not publishable, or we did not find any journal where we can put it, and then we, anyways, made them public on GitHub or other sources.
Mm-hmm. Yes, this is an interesting form of collaboration, because I think for students who are only starting out, the most obvious one is when professors are interested in topics of each other, they find a common means, and they can develop something together. But there's a different way to collaborate inside communities when just many people recognize the need in a tool or a resource. I wonder, Julian, if you could also talk from the perspective of experienced scientists, and what motivates you to join and work within communities, and how does it benefit you?
So as I said before, basically I'm in several of these communities for real some time. So I was really happy that I joined some of them as an early PhD, because that was the way to get out of your silo, really, to meet persons around the world. And I really think, at least in Europe, I know a lot of proteomics bioinformaticians now from all the different labs, and I would not have met them before, and it's that is basically the most interesting thing, that you really know all of them, you know what's going on, you know whom to ask, and also can learn new aspects. And then obviously if you go on and on, for me now, networking is still very interesting when I'm writing grants now and things like this. So that's the important thing you do after PhD and after postdoc, I guess. But still, it's very interesting to learn these things. And also, basically, if you think of looking for new opportunities, or if your PhD is over and you want to join another lab, it's very nice if you already know a lot of the community and maybe know to ask, or just ask for letter of recommendations and things like this. It's also a very, very good thing.
Fantastic. And
besides the science, that is obviously, so the science is also very good there. Just working on very interesting topics.
I guess it can help to go out from your bubble too, because sometimes scientists are very into their topic all alone, but then you can get a different perspective from many minds. And do you encourage your students to participate?
Yes, I do. Actually, that's what they blame me for. Basically, was the first thing. So the students I worked with together, my first group started during Corona, so it was a bit hard. But the first thing I told them was, you go to this conference and join actually this group. You don't only go to this conference. And they stayed. That's good. Some of them stayed.
Yes, I hope they recognized a good side of it. Olga is writing in the chat, thank you a lot for information. I hope I will use it in nearest future. I hope so too. Also, Amina, thanks for helpful information, and Irina, thanks for the lecture.
I think Mikhailo still has a question.
No, sorry.
Okay. No problem then. I don't see that anyone has any discussion points right now. So I hope you get a little bit inspired. And I just want to acknowledge from the side of person who went through Ukrainian academic system and then went to the international ones. Sometimes when you're starting out, you can feel powerless as a student. Like, you cannot lead anything, you cannot decide something for yourself, though you're really motivated to put effort into science that is around you. But here with this lecture, I hope we managed to open a window for you to show that you can make a difference. You just need to want and come join the groups of people who share your interests. Then thank you very much for your attention, and thanks a lot, Julian, for your support and contribution.
Thanks for having me here. And have a good, nice day, nice evening or day, whatever comes next to you.
We'll stay in touch then.
Yeah.
Good day.
Thank you very much.